The word omics came from the suffix "om", which means group of or a collection of things, and the suffix "ix", which means the study of, the science behind, or the discipline. In this sense, omics means the study of different groups of biomolecules that makes up a human being. It studies the background processes that keeps us alive. The central dogma, in the simplest sense, states that DNA replicates itself, DNA makes RNA, and RNA makes protein. It is a linear process. DNA is made up of base nucleotides, adenine, guanine, cytosine, and thymine. They are paired specifically, the purine adenine with the pyrimidine thymine, while the purine guanine with the pyrimidine cytosine. Together, they form a sequence. This sequence can hold the genes, or the instruction manual, and also the non-coding parts that protect and regulate the DNA itself. With this, genomics is the study of the entire gene set of organisms. Not to be confused with genetics, where genetics focus on a single gene's function and composition. Genomics, on the other hand, looks at all the genes and look for their relationship. Genetics is a subset of genomics. In genomics, these are usually the questions to ponder. Next in the central dogma is the RNA. It is a single-strand copy of the DNA. However, unlike the DNA, there is no thymine base. Uracil replaces thymine. RNA serve multiple functions in the body, as in like messenger in mRNA and transport in tRNA, to name a few. mRNA holds the codes on what proteins to produce. rRNA and tRNA aids in the translation process. srpRNA, or signal recognition RNA, handles the member integration function. There is also one RNA that is specific to bacteria called tmRNA, or transfer messenger RNA. It acts as a rescue to stalled ribosomes. Wikipedia has a good list of RNA roles and functions. The byproduct of transcription is called a transcriptome, and transcriptomics is the study of set of RNAs in an organism. These are usually the questions for transcriptomics. In the last stage of central dogma, RNAs are translated to synthesize protein. RNAs are read by sets of 3 nucleotides called codons. An amino acid is formed per codon. Proteins are made up of long amino acid chains. Muscles are not the only proteins that we're talking about. There are actually 4 types of proteins that have different functions too. 1. The globular proteins have a spherical shape. Examples of these are enzymes, antibodies, hemoglobin. They are called biomarkers because their existence in our body may indicate an abnormality. 2. The fibrous proteins have no shape but are linearly and elongated in form. Therefore, structures and support like in our muscle tissues. 3. The membrane proteins have no shape but are found within or attached to a cell membrane. 4. The disordered proteins have no specific structure nor shape and their function depends on it. Often, the drugs that are being developed targets the said biomarkers. Since proteins are kind of vast, these are usually asked by the researchers. Going out of the central dogma, here are the other branches of omics. Have you ever seen a plant of the same species, probably of the same parent, but grows differently? A basil plant growing indoors are relatively smaller than that of those grown outdoors with all the sunlight. Same genotype but different phenotypes. This is called phenotypic plasticity. There are no changes in the gene nor the genome, but they vary in how they look like. These instabilities are puzzling to researchers and challenges how we understand genetics. Epigenomics studies all these mutations of genes and variations of gene expressions. It takes note of the effects of environment, diet, and everything that will alter DNA. The prefix epi added to genomics means more than that of the genome. Mutations in the DNA, like in DNA methylation, which is a biological process of attaching methyl to a nucleic acid base, are observed in epigenomics. Epigenomics takes into account the effects of these changes to the genome and to the organism. It looks how permanent the changes are and if they will be passed to other generations. The food we eat gives us the metabolites we need to survive. Metabolites are vital biomolecules that have low molecular weight. They are products and byproducts of biochemical reactions. Lipids, carbohydrates, and enzymes can be acquired through our food, and our bodies can also synthesize these. Vitamins, on the other hand, are processed and generated by other microorganisms that live in our body. This will be discussed more in microbiomics. Metabolomics is the study of different metabolites and their associated chemical reactions. It keeps metabolic data and combines them with disease data to understand the epigenome. We don't live alone. We may not see them, but they're definitely always with us. Since time immemorial, microbes have lived and coexisted with us humans. Some microbes may be scary, but these microbes are beneficial to us. We are in mutualism with them. We eat the food, some of it can be digested and absorbed by the system, while some cannot be done alone. Bifidobacterium species in the gut microbiome are good at synthesizing or producing the B vitamins such as biotin, folate, thiamine, and niacin. These B vitamins are essential for our metabolism and maintenance of cells and tissues. Microbiomics is the study of an organism's microbiome. A microbiome is a collection of naturally occurring microbial community, their genetic material, their structure, their function, their dynamics, and their effects in human health. A change in homeostasis with the microbes changes a person's health, and in turn, a disease may be brewing. Based from our discussion, we can graph the connections of each branches of omics. Genes from genomics are transcribed through produced RNA that is being studied in transcriptomics. Genes in the genome varies in expression, and environmental factors affect the genome. This is studied in epigenomics. Epigenomic changes in the genome also affects the transcriptome. A change in transcriptome leads to changes in proteome. Since histone is a protein, alterations in histones must be taken into account by epigenomics. The metabolites work with the proteins, and the change in the proteome also changes the metabolome. The food we eat may also cause mutations in our genes, which is part of the epigenome. The microbes in our bodies synthesize the vital molecules in our metabolome. The omics discussed previously only looks at single organism. In this world, we live with other organisms as well, and they have an impact in our system. Metagenomics takes a sample from the environment and identifies which organisms exist by identifying their genome in the pool of genomes. It also aims to study similarities or commonalities of different places by studying the existence of their genomes. Metatranscriptomics profiles the gene expressions rather than the gene itself. That is the job of metagenomics. It consists of changes in expression profile from the community. With this, the functions and activities of the transcriptomes are observed. Metaproteomics takes all proteomes found in community. It studies the metabolism and physiology of each organism by looking through the proteomes. Handling enormous data is painful. Storage and retrieval extremely difficult. Finding connections is nearly impossible when done manually. Accessibility to the data is another problem. The creation of database management systems solve this error. Here is a superficial look on what databases can do and how it can help omics data. These are some databases for each dataset. Say you found an unusual sequence of nucleotides and you want to find related organisms that may share the same or similar sequences with the data at hand. An appropriate blast search in KEGG, NCBI, and EBI databases may give you hints on what it could be. Like when the SARS-CoV-2 was still called novel coronavirus, no one found a perfect fit for its genome, hence the name. This genomic data can also lead the way on finding the right ancestors in a phylogenetic tree. Clinical Genomic Database of National Human Genome Research Institute helps biomedical researchers by curating statistically and medically significant genes that has known somatic effect. The databases listed will be of great help if you're looking for pertinent omics data and their specific function in the body. In these difficult times, one human behavior puzzles me the most. Why are we inclined to help others in need rather than preserve ourselves? This is called altruism. Quoting from OpenText BC, altruism in social psychology refers to any behavior that is designed to increase another person's welfare, and particularly those actions that do not seem to provide a direct reward to the person who performs them, helping without asking for anything, or selflessness. Being taught to be kind may be one of the reasons this exists, but in a 2013 study from Zaki and Mitchell, they noticed that these behaviors are almost always automatic and inert. Now, since the study said that this behavior is ingrained to humans, what is in the human system that exemplifies altruism? Going to NCBI, you can actually search for the word altruism. EBI also supports this feature while KEGG does not. Sifting through the filter, you can set it to gene to be specific. In here, you can see that there are currently 4 genes associated with altruism. AVPR1A looks interesting. Let's open it. NCBI, like other genomic databases, will show you useful information about the gene. Its symbol, name, purpose, and of course, the summary. You can even find orthologs of AVPR1A. An ortholog is a direct descendant of a gene with the same biofunction. In some cases, paralogs or duplicate with different function may be given too. You can download these data to further your studies. The data are in FASTA format. Alignment, search tools in local and wider contexts are also available. Opening the gene data gives us the ID of the gene, the name, the organism where it's found, which chromosome is the gene located, and lastly, the whole sequence of AVPR1A. Next up, the RNA sequence of AVPR1A is as follows. Lastly, the amino acids synthesized from the RNA can be downloaded too. Browsing EBI and searching for the proteins associated with AVPR1A gives you its relevant proteins, shows you the different functions of the protein. It also includes metabolic processes associated with the gene and the protein. EBI and NCBI uses different literatures to connect the epigene of AVPR1A. Here you can see the gene expressions based from different studies collected by EMBL EBI. GEO or Gene Expression Omnibus datasets of NCBI also does the same thing. Other epigenomic databases like Safari Gene takes note of the mutations and associated disorders. It also includes the publications that support the claims. Microbiomic data are quite difficult to find if you have a specific gene in mind. However, looking at the bigger picture, we can look for the effects of microbes to the altruistic tendencies of organisms. Like in the example, one example shows that there may be a negative effect in the prosocial skill of mice if antibiotics are given early in life.